more of a journal club than it's a talk. So since my biology work is all in neuroscience, I didn't think you want to hear about this. But I picked up this topic because, frankly, I was asked to review a paper, and I now seem to be sort of the go-to reviewer on this for several journals. So what is magnetogenetics? And the story begins with a couple of papers in about 2015. But the basic idea is that what if you could tag a magnetic particle and get it to bind to an interesting protein, in this case a membrane protein, which is a thermally sensitive one, and use it to switch that channel open or closed, right? And this was basically reported in this paper. But the idea, I think you can see, is right here. If you look at the, is there a pointer somewhere?
It's right there on the ledge.
Oh, I'm blind. Thank you. Right. So the idea is to find a little magnetic nanoparticle, and this is one. And actually, the nanoparticle is one that's throughout our bodies. It's ferritin. Ferritin is the iron molecule that is used to store iron in the body. They played a nice little game by taking the iron molecule and gluing it onto this protein. And you can build a sort of a nice tagging system by which, in principle, you can inject this, have it dock to something on the molecule, and therefore you can stick it around. And then after you've done that, you apply either RF or a magnetic field, and you open up, in this case, a calcium channel to do that. So that's the idea. Quite a bit of work to show that it actually works. I don't want to spend a lot of time on this, and also partly because I don't understand enough biology to be able to describe this properly. But there are a bunch of things that you do. This is the one that you think will work. You can also get the ferritin to stick to the membrane, or you can just disperse it everywhere. This is just an assay. Probably you can't see it very well, but the point is because you've also got GFP and various other molecules stuck on here, you can check that the things are where they're supposed to be, and that sort of works. Then the next one was an experiment that they did with mice, so this was aimed to control blood glucose. So basically you inject a mouse, you wait for it to get expressed, and then in this case you microwave the mouse, gently microwave the mouse, I think, to do this, right? And what you can see, according to this, if you believe all of this, is that blood glucose in this case is red. So the one that you wanted to do, that you believe should work, is the green one. The blue is one of the controls where there is some ferritin around, but it's not necessarily linked to the channel protein, and this is the control. Done about half a dozen times. It's plausible at some level. But that was RF. This is essentially the same experiment now done, but with a magnetic field. And again, here it is. So you're on for a time of about 60 minutes in a magnetic field. And these are the two cases. One of these things, well, they're basically both with the ferritin in, but it's sort of with and without the magnet. So that's all very nice. But then the question comes, okay? Yeah, maybe it works in practice, but does it work in theory? Right. So this is by the way, this is a quote typically about Chicago economists, right? That we do this because the kind of idea that's proposed is I have a little magnet in here. I'm applying a magnetic field. I can align the magnet to the magnetic field. It tugs along the molecule and moves it there. No, that's a nice story to tell, but it's actually hard to make sense out of that. And that provoked a rant from Marcus Meister, actually a rather useful paper, better say. That basically he just says, it's 5 to 10 orders of magnitude away from being a plausible mechanism for what's happening, and therefore throwing some doubt on all of that. And so, what's the critique, basically? I'll talk a little bit more about ferritin, the molecule itself, but actually ferritin is I should say, it's something that grows a nanocrystal of ferritin inside some package, sort of a 20-former holding all of this thing together, and you gradually sequester iron oxide in there until you build up a nanocrystal. The nanocrystals, when they're fully formed, are all pretty much identical. They're about 4,500 iron atoms, but they're not ferromagnetic. They're actually antiferromagnetic. So what does that mean is that there's a very strong magnetic interaction between the ion spins, but the ion spins are basically anti-parallel with each other. So I have an ordered state, but it's not a magnet. I mean, it's not. It turns out to be lightly magnetic because, of course, the crystal has a surface, and on the surface there are some uncompensated spins. So it turns out that the magnetic moment is about n to the 1/3, where n is the number of particles. So that's about 20 ion spins. That gives you 100 Bohr magnetons. And so if you take this number, compare it to kT, it's only about 20% even in 1 Tesla. Okay. Furthermore, if you start computing the force that you could get by taking this magnet and trying to orient it in the field, it's going to produce a force of the order of about 10 to the minus 23 newtons. So we've seen forces measured in this talk, piconewtons you're proud of, right? So that this is kind of a long way off from being really plausible. Now, something even worse than that is that actually ferritin is what's called a superparamagnet. So I said it was magnetically ordered, so the bulk material orders are around room temperature. But ferritin is so small that in this kind of paramagnet, it turns out that there are two equivalent states. I can have spin up, spin down, or spin down, spin up. And so there are two possible ground states, like a regular ferromagnet, where I could point the magnet up or the magnet down. And the system is small enough that it thermally fluctuates between those two states. It's called a superparamagnet. I'll show you some more about that. Okay. And at room temperature, it's actually fluctuating on a scale of nanoseconds. So there's no plausible way, again, that the little magnet stays pointing in the right direction for a persistent force due to a field. So that exercised Marcus and a few others. I particularly like his rant here, which is about this. He said that I'm not sure whether this is fair at all. I mean, I always tell people that if you've got experimental data, you should publish it. And you shouldn't have to have a theory in order to get it there. But as you say, he was, because this paper and a couple of similar papers caused quite a fuss. There were review articles written all about this. And Marcus said, what's the community doing? As he says, claims that violate the known laws of physics often turn out to be wrong. Right. So does it, right? So I want to spend the last piece of the course saying, having the last piece of the talk saying, is there something plausible that could explain this? Assuming, of course, that the experiment's actually correct. I think last year, actually, there was another paper published with by some of your colleagues, actually, with I think a good chance of being the right mechanism, although I think not expressed in exactly the same way. But in particular, they repeated some of these experiments, but I think under much more controlled lab conditions, and agree with it. But they also pointed out that there are other effects that can take place in a magnet, and in particular, that if you can change the entropy associated with the spins, then there's a lot more stuff to be had. So let me take you through that a little bit and a little bit more about the microscopic story. Okay, so as I said, Ferritin is an ion storage molecule. It's in everything. It's pretty much deeply conserved. It's all the way from bacteria up to us. It has a protein shell, and it contains about a 7.5 nanometer core of something which is actually a well-known mineral, or a version of this. It's called ferrihydrite. If you ever see a red stuff leaching out of a sewer or something like that, assuming that it's actually the red is iron, it's typically this molecule. So it's rather like hematite, but hematite is just Fe2O3. Hematite doesn't have any water in it, and this has variable amounts of water, but this is kind of the official composition. Okay? But it's a pretty robust antiferromagnet as in bulk. So that means that the spins are all locked together in an antiferromagnetic configuration, and it's got a tiny ferromagnetic moment, but that's a finite size effect. So I said there's a mineral, you can do this. And then the other point to realize is that this order flips as a function of time. It's a superparamagnet. Okay. So let's see if we can get some handle on how much magnetism there really is in this material. It's not easy to do because, of course, if you've got nanoparticles, you can't put a squid on top of it and try and figure out what the magnetism is, especially if it's fluctuating. But one way of measuring this is a very old effect called the Coton-Mouton effect, measured in 1907, that just says that if the refractive index changes with the field, right, and the theory of this was done by Néel, but simply you expect this coefficient to depend on either the change in the susceptibility or the change in the magnetic moment. It doesn't matter either of each of these things. And this group, actually, for different reasons, studied nanoparticles of this material, and you can make it in lots of different kinds of ways, and that's sort of interesting. So actually, this is bacterial ferritin, actually, which is down here. Everything is sort of nicely quadratic in the field. These are other nanoparticles which also contain the same mineral, but they're made in different ways. I think some of these things are things that you buy over the counter to improve your iron and can swallow and drink, and so you see all of these effects. And on the basis of that, you can use this to determine either the susceptibility or the moment. And in particular, you can also look at what you would expect if the effect is not entirely linear, and the one where you see it the largest is an iron sucrose solution here. And in that case, you can actually get fields which are big enough to saturate the birefringence, which means that you really are lining everything up. You're a very long way away from doing that in this material. I think it's not really understood why it is that there's much more moment on these particles than there is in ferritin. Now, the next thing is that this isn't the first controversy involving ferritin. I said the spins flip up and down. Okay. Of course, that's an activated process, so as you go to lower and lower temperatures, you'd expect it to stop. Well, it doesn't. It actually keeps going. This was proposed back actually in the 90s, that what you were seeing at very low temperatures is actually quantum tunneling of the moment. I don't want to go further on this, but it meant that the topic got well looked at by this. And actually, this remains pretty controversial about whether this was really happening or not. But there's quite a lot of data on the superparamagnetism from this paper. I'll show you, and let me just turn that up because there's some useful things here at high temperatures. So what do I mean by saying that it's a superparamagnet? It means that you measure the susceptibility as a function of frequency. If you measure it very quickly, the molecule is frozen. If you measure it very slowly, the molecule has time to flip, and therefore there's a resonance somewhere in between. And so this is looking at the susceptibility at different frequencies from 100 hertz to 3 kilohertz, and you see there's a peak which shifts as a function of temperature. This is the so-called blocking temperature, and you can plot the blocking temperature as a function of frequency, and if you add to this much higher frequency measurements that you can get from Mossbauer, you can plot this all of the way up here, and this gives you a characteristic frequency, which is some rate times this, and this is, I say, about 10 picoseconds at room temperature, because the activation energy scale is about a little over 300 Kelvin. Now, this by itself is potentially interesting, because what you see is that even as you're getting up to room temperature, the characteristic frequency of this flip is coming on to an interesting time scale in terms of any kind of biological process. This is the kind of time scale on which things actually move. Furthermore, as I mentioned, and it's not quite clear about the microscopics of this, it's also at some point when the temperature gets up to this scale, you'd really expect the antiferromagnetism to go away, and you expect to go from an antiferromagnet to a paramagnet. And in fact, the bulk mineral has a transition somewhere around room temperature. Okay. So the suggestion there then is that what's actually happening here is not that you're doing any mechanical force, but actually you've got an inbuilt refrigerator. Right, so what do I mean by that? Well, there's a thing called the magnetocaloric effect. So this is actually used to build refrigerators, and it takes you around a thermodynamic cycle. And this is drawn for a ferromagnet, but I'll put it up here. So imagine that you applied a magnetic field and aligned all the spins. Then, if you take the field off, and the system was not supposed to be ordered, it was supposed to be disordered, it's going to find its way to a disordered state very quickly over here, so the spin's disorder. But that's happened very quickly, and because it's happened very quickly, that's an adiabatic transition, and so there's no heat coming in or out, and therefore you have to balance the entropy change with some internal energy change, and the system will get cooler. So you switch off the field, it gets cooler, and now then you leave it to sit there, and of course it'll gradually warm up, it'll absorb some heat, and you'll get back to the operating temperature. And then of course you put the field back on, and you move over here, and the system is now in the disordered state, whereas it should have been ordered in the field. So in this case it must warm up. And then you allow it to come to equilibrium with the environment, and it ejects the heat to a heat sink. And you comes back that way. So this is the way refrigerators work, or most refrigerators work. You know, the garden variety refrigerator, of course, the phase transition is not magnetism, it's liquid to gas. And the field you turn on and off isn't a magnetic field, it's pressure. But nonetheless, the same principle can be used at any phase transition. And that, I think, really was the proposal in this paper. But it's got some few problems, right? So the nice thing about that is if you are able to induce this phase transition, there's actually quite a lot of entropy. Because the entropy now, you know, it's not relying on all of the spins that are actually lined and pointing in the external field direction. It's all the spins in the crystal. The entropy is of order kT times the log of the moment. And this will typically then, in an adiabatic change, result in temperature changes of the nanocrystal, which may easily be tens of Kelvin. This is how these things work. Okay? And furthermore, usually there's a, you know, particularly for an antiferromagnet, there's always a strong coupling to elastic effects, and therefore the crystal can change its shape. So going through this, you would also have a shape change. However, in order to do this, it requires a refrigeration cycle, because of course, you know, imagine I've got a little nanoparticle in here, I'm applying a field. Yeah, the first time I do it, it cools down, but then if I leave the field on, nothing else changes. I actually have to take the whole thing through a cycle. Okay? So I'll have some kind of cycle like this, and this, of course, would be the temperature change. Heating, cooling, and I will need, you know, enough time to equilibrate with the environment, so this has to be long enough. But it's also true that I don't change the average temperature of anything. Right? So you're either absorbing heat from the environment or giving it back to the environment, so you don't change the environment at all. And furthermore, you'd actually need really good contact between the protein and the ferritin. Good. And poor contact with everything else. Because even though I say I may get a temperature change of 10 Kelvin or so associated with this little nanoparticle, it's in a bath, right? And it will very rapidly equilibrate to that, and therefore it needs to be kind of embedded in the molecule so that it's preferentially operating next to that. If I look at magnetoelastic effects, again, thinking about this small molecule, if there's even a few percent change in the shape, which you can easily do, that in the end is probably no more than a displacement of about 1/10 of a nanometer, which isn't small, but potentially could work. And of course, there's no evidence for a nearby phase transition in ferritin. Now, so I think one is still confused about what might actually be happening in the system, if indeed there is something there. I sort of begun to be convinced that there is some effect which isn't a placebo effect. I mean, a placebo on the experimentalists rather than anybody else, right? Not the mice. But I will point out, actually, that there are plenty of materials with really giant effects that you could harness. So this is one of my favorite ones. This is actually a manganate material. These things were fashionable about 20 years ago for something which is called colossal magnetoresistance. So this is actually resistivity on a log scale as a function of temperature. And at different magnetic fields. Now, this happens to be a low temperature one. You can make these things happen near room temperature with very substantial shifts of the transition, and there's a huge amount of entropy here. And also, there are other things that you could find useful, like a metal insulator transition that goes along with it. Okay. It seems to me to be not impossible to imagine that maybe not with ferritin, but maybe with other materials, if you can find a way of making them, biologically, getting them biologically into the body, or you could tune things to make them operate in certain ways. There is potentially, I think, enough magnetic entropy in these materials to have a substantial effect. And that actually is where I wanted to stop. And see if somebody else has got a better idea, by the way, about how this might work. Thanks.
Okay. Questions, comments? Yes, Jose.
If the tunneling effect, how many spins are you flipping?
All of them.
What?
All of them.
And they're what?
Oh, so the 4,000.
4,000.
4,000 spins.
Compute the probability.
Well, as I say, again, so the interesting thing is, this is another place I say where theory and experiment don't agree. It seems to me to be a very large number to actually get a decent tunneling rate. But as you know, computing tunneling rates is a serious challenge. But experimentally, it is true that as you go down to low temperatures, below 10 Kelvin or so, then the magnetization certainly continues to fluctuate. It's no longer activated. And there are a few other aspects of it which are considered.
Or some things moving around.
Well, right. So again, now you can get into details and say, are they all tunneling? Could there be some defects? Could some of them be there? That's been occupying people for a while.
Peter?
I kind of lost a little bit of track through the very fascinating theoretical discussion about what the experiment was. In the sense that I can easily imagine that you can put ferritin-coated genetic particles that go somewhere specific.
Right.
Just because they're foreign, they do something. It seems like the experiment's supposed to tell us that something's different when they're done with a magnetic field on than when the magnetic field is off. That's what I missed
Right, so so actually, on here, so this is regulating glucose, right? That's the, right. So if you like, there are many experiments in here, but this is sort of the key one. If you like, you inject somewhere back here. This is, one of these curves is with the magnet, one of these curves is without the magnet. Right, so everything else is the same, and you're just measuring the blood glucose level in doing that. But no, there's a few other things.
So something does happen without the,
Yeah.
without the magnet.
Right, right. As I said, there's
So
I mean,
it's kind of an enhancement. But that might suggest there's some kind of more macroscopic thing going on, like
Well,
you turn
yeah.
the magnet on and it just
Well, I mean, but
go
actually
through the
notice that
needle or
you've got
something
an
like that.
anesthetized mouse at that point. So you might expect something to happen there. I mean, this one that I showed you I didn't talk much more about, it's actually a little bit easier to believe in terms of mechanism, is where you actually treat with RF, right? And in this case, again, they've done actually quite a few cell level tests to demonstrate that they can, so these, sorry, I think I probably must have been on a previous, or maybe I left something off here. Sorry. Right. So these are different cells which have gone through somewhat different protocols for all of this. So this is not a whole mouse. And again, you have to believe that the control is here, the magnet is there, and this is, what is it looking at? Sorry, it's a change of, I think that's actually calcium, which they measured there. But by the way, I mean, there's all kinds of ways that these things can go wrong. I think you know that.
My question is close to the one of Peter. I must have missed something. In your title, it was magnetogenetics, and blood glucose and other things are exciting and interesting, but doesn't seem to be genetics.
Oh, okay. So the reason that this is genetic is right back here, is that these things are tagged. Right. So you can choose. So you have a ferritin with a tag, and you can make that genetically specific to bind to different places in the same way that you do with the optogenetic molecule. Right. So take your optogenetic protocol, replace GFP by ferritin. That's the sales pitch. Right. Yeah. Sorry. Well, I didn't say it very clearly, but that's why it gets people's attention, right? So
I had two questions.
yeah.
I mean, if the goal is to just why not just put a ferromagnetic particle in? Why are you doing it in the case where the effect is going to be smallest, except due to surface effects? And the second question is, what strength RF are you talking about? Are we supposed to go back to worrying about cell phones again?
Oh. Okay. Fine. Right. Okay. So firstly, right. So look, obviously, the right thing to then you get to birds and seagulls and whatever things like that. Right. So if you wanted to run the numbers for a regular ferromagnet and say how much magnetic moment do I need to have this, I would need a pretty sizable magnetic particle to be able to do this in.
So magnetite, the type you have in bacterial cells, is not going to do anything.
It's not actually going to do anything, right? Okay. So, and then I think for that reason also, as I say, I think that this is still, RF at the level of cookery rather than our RF at the level of cell phones.
Okay, so we can't use our microwave oven, but we can still use our cell phones. Got
Yeah,
it.
that's right. Well, you can't climb into your microwave. I think that's right.
Okay. Any other questions? Okay, if not, then we'll thank Peter again.
Thanks.